Hello, welcome everybody to the session on the future of bioprocesses in biorefineries and how to scale up more resources. My name is Lewandowski. I'm holding the chair on bio-based resources and the bioeconomy at the University of Hohenheim in Stuttgart, Germany. And there I'm also steering as the bioeconomy strategy as Chief Bioeconomy Officer and a spokeswoman of the European Bioeconomy University. We will have in this session 4 presentations in the field of the future of bioprocess and biorefineries, and there will be very interesting 4 presentations which will be covering the presentation of innovation platforms, company activities, projects, and the zero concept in the bioeconomy. I assume that keeping the 10 minutes presentation time will be challenging, but I would like to ask all speakers to do so, as there should also be room for discussion. And I would like to motivate all participants to write their questions into the chat, and I will try then to bring them to the attention of the speakers. The first presentation will be given by Matthias Slatner. He studied biotechnology at the University of Life Science, Boco in Vienna, Austria, where he also did his PhD in the field of enzyme technology. Presently, Matthias Slatner is Innovation Manager of the Austrian Center of Industrial Biotechnology, where he's responsible for the open innovation strategy of ACIB or ACIB. We are looking forward to your presentation on the development of an open innovation platform to connect biotechnological research and business. And now the floor is yours.
Thank you very much for the nice introduction. And thank you to the EFIB organization to invite me to hold a short presentation about our digital open innovation project. Please allow me to say a few sentences about ACIB, the Austrian Center of Industrial Biotechnology. ACIB is a multidisciplinary research center for biotechnology with the goal bridging the gap between academic research and industrial success. Our key disciplines are biocatalysis and chemoenzymatic synthesis, enzyme technology, cell line development, bioprocess technology, bioeconomy, and also the field where I'm active in, environmental biotechnology. We are about 200 people working in Graz, Tulln, Vienna, and Innsbruck, and our owners are 5 Austrian universities, and ACIB is partly funded from the Austrian COMET system. How did our digital open innovation project start? Of course, ACIB as a research institute uses quite a lot of digital techniques. So we have, for example, a digital communication platform for our employees. We will be also hosting a European Researchers' Night. And I, from my own, have been asked to create an open innovation office, a physical office in our site in Tulln in Lower Austria. And then very suddenly, due to the Corona crisis and the lockdown, there was some time, there was some space, and I started to develop a digitalization step to go from the physical open innovation office to a virtual one. Why in Lower Austria? Lower Austria is one of 9 Austria provinces, and we have quite a lot of research institutes here, of university institutes that work with biotechnology, and we have a lot of companies in Lower Austria who are doing their business with biotechnology, and we found out that there is not that much communication in between researchers and companies, in between researchers and researchers, and that also for small companies, small medium companies, startup companies, they have sometimes problems to access research results, and this was the step we wanted to drop in and to prepare a better connection by introducing a physical office located in the center of Lower Austria in Tulln. Our open innovation approach is that we want to collect ideas coming from students, from the crowd, from interested people, to develop with these ideas some business models to go to the market and to combine them with our experience with investors, with ideas from our employees, with legislation part to create, in the best case, new business opportunities. The idea of our digital open innovation platform started when I found out, after operating 1.5 years, 2 years, the Open Innovation Office, that there is really a demand to discuss ideas with experts. And a lot of people do not have the best possible access to discuss with experts. So the idea was to design something like a Ask an Expert page, homepage, digital possibility to go in direct discussion. But I found out that maybe this is not enough, and so we designed a 4-step innovation platform with Ask an Expert, Virtual Idea Lab, Co-Creation Center, and a Biotech Entrepreneurship Toolbox. So the first part, and for me also the most important part, is the Ask an Expert button that will be online on our homepage about in the middle of November. Here it is an offer to interested people to ask questions, to go in direct contact with researchers. So it is of course free, completely free and available for everybody, and it gives a chance to post questions or to post ideas, to get a contact to experts in biotechnology, to discuss them, to get answers, and practically it will be the questions will be sent to our business development team. We are 3 people in our team, and if we can answer them by our own, of course we do so. If we cannot answer them, we have a pool of around 200 people working in various areas in biotechnology who can give answers to questions. From this point, the second step, and I wanted to visualize it with a nice photograph from a vacation in England with Stonehenge, to create a virtual idea lab. That means that's the real open innovation point, where ideas can be posted and can be open, discussed with expert and with other people who are interested in these ideas in an open way, where everybody has access to the discussion. Of course, I know that there is always the conflict when you speak about open innovation. There is a conflict also with secrets, business secrets, IP rights. So at one point, when the discussion gets very much into detail, or when the discussion really enters a point where business secrets are communicated, there will be the possibility to go to a closed area to discuss ideas not open for everybody, but in a closed center for registered people. And this will be provided by ACIB on our homepage. As the last part of our open innovation platform, I tried to develop an entrepreneur toolbox. In Austria and probably in all European countries, there is quite good possibilities for people who want to make a company, who want to do a startup, to get advice in many aspects of business. But biotech business is sometimes a little bit different. And so I try to accumulate their ideas or information about funding startups in the biotech field, practical ideas, probably some webinars, some documents to download. And I hope to encourage people who have the idea to found their own business to use the information we can provide there. I did a first trial together with the university in Sarajevo to give a lesson in biotech entrepreneurship, and it has been a big success. And I want to use these ideas to put it also on the open innovation platform. Well, we were very happy to get the project to host the European Researchers' Night in Graz. But then we found out that we have to make it totally digital. And this, of course, fits also to our open innovation strategy to learn how to digitalize such events. We are quite experienced with giving workshops, to making events for children, for students, to get first ideas about biotechnology. But this has always been practical, physical, and it is the first time we do it digital. Our key topics are food technology, health, and renewable resources. And we try to make a nice program with online discussions, game quizzes, presentations, open lessons, and do-it-yourself experiments to interest young people for life science. To summarize the idea of our open innovation platform, it should be a tool to develop new ideas and an approach to integrate the crowd to biotechnological research. Of course, it should help networking, connecting people, connecting researchers and companies. And it should also show that ACIB is in a very small area, also working as an accelerator, because we have something like 10 startups that already came out of our research groups. I want to thank our team that helped me to design this open innovation platform. And I was in summer very much surprised to get the Innovation Award for our idea. And of course, I also thank the province of Lower Austria for providing support to the Open Innovation Office and the Open Innovation Platform. Thank you very much. And I'm happy when there are some questions.
Yeah, thank you very much for your presentation. Also, congratulations with the Innovation Award that you got. I think you deserved it because it looks really interesting. And I wonder how, with this rather small team of people, you manage all this. So I have seen so far no questions in the chat. I see somebody is writing. But I would like maybe start with a question by my own. It looks very interesting and appealing to participate somehow. But who could participate? To whom can you be open? Is this more an Austrian activity, or would you also be happy to invite people from other from other institutions?
Yes, of course. We are an Austrian company, but we are working in the field of biotechnology with international partners, and there are quite a lot of our employees who are not working in an Austrian lab, but somewhere in Europe, and we even had projects with overseas locations. And of course, we try to provide a network of biotech researchers, mainly in the field of our key disciplines. And the idea of the Open Innovation Platform is when we get an interesting question that is in going to details that cannot be answered easily, we would like to look in our portfolio of researchers who has experience in this field and to forward the question to her, to him, to connect the person who is interested with the researcher that can give the best question. Of course, it is not limited to Austrian people.
Mm-hmm. Yeah. Yeah, that would also have been, I think you answered the question that Yvonne was posting also.
Yeah, yes, I just read it.
You just read it. So I think you have already answered that question. And I think I was asking myself a little bit when you were presenting, isn't it a challenge to motivate or to get people and experts to help answering these questions, because mostly scientists have a lot of things to do, and if you approach them, they don't always have always capacity to answer that. Does it work well, or how do you motivate them?
Of course, you are completely right, and just in a position as business developer, I know very much about this problem, and I think the idea from the Ask an Expert is that that our team of 3 people, and we are all biotechnologists on our own, we can somehow create a funnel and to take the question and provide it for the scientists, that the scientist is really interested also to give an answer. So I want to avoid that scientists are bothered with quite simple questions that we can answer on our own, but when questions we will try in our small but existing business development team to find a kind of business case behind it that makes it interesting for the scientist also to drop in to this question. And we are quite experienced with this, and also until now we were able to develop quite a lot of interesting ideas, especially not to typical fields of biotechnology, but to areas where people did not think that biotech could give an answer to their questions.
Okay, yeah, sounds great. Maybe I will contact you sometime as well. Thank you very much for this input. And yeah, I think it's time to come to the next speaker. And then at this point, I would like to introduce our next speaker, who is James Gaffey. And he is a principal investigator and co-director of the Circular Bioeconomy Research Group at Shannon Applied Biotechnology Centre at the Institute of Technology, Tralee in Ireland. And amongst others, he's also the scientific coordinator and principal investigator of the BBI joint undertaking ICT Biochain initiative. And he's also principal investigator on the BBI BioSwitch project, and that is the project from which he will report now in his presentation. And I'm looking forward to your presentation, James.
Thank you, Iris, and thanks for the invitation. So as Iris mentioned, my name is James Gaffey. I'm co-director of the Circular Bioeconomy Research Group at Institute of Technology Tralee in Ireland. We're a multidisciplinary group. We have expertise across biorefineries, green chemistry, biotechnology, business models, and sustainability. And we participate on a lot of different European funded and nationally funded projects, from research and innovation and demonstration actions to desk-based research activity and coordination support actions, as Iris mentioned, one of which I'll touch on today. We're also active in education, so we co-design and co-deliver Ireland's first postgraduate taught program in bioeconomy called Bioeconomy with Business, which is a collaboration between ourselves at IT Tralee alongside University College Dublin and Teagasc. So I'm going to talk about the BioSwitch project. It's a BBIJU coordination support action. It's coordinated by Click Innovation, who are a Finnish bioeconomy cluster. And the focus of BioSwitch is on brand owners who are a key stakeholder in the bioeconomy. The focus of the project is on supporting brand owner uptake of bio-based products, particularly replacing incumbent fossil-based products with bio-based alternatives. Brand owners can provide, penetration of mass markets for bio-based industries, bio-based ingredients. They can also be key influencers over consumers, particularly for an area like the bioeconomy, where lots of new products with new functionalities are coming to the market. It makes it even more important. So we haven't long kicked off. We only kicked off in June, and I'm going to touch a little bit on our methodology, but also present you some of the results of a survey which we're just in the process of finalizing. So since June, we've been engaging and building up our brand owner networks, and we've been working with them, interviewing them and surveying them to get a better understanding of the motivations, perceived risks, incentives for brand owners making a decision to switch to bio-based. It could be bio-based ingredients, could be bio-based products, could be bio-based packaging. And we're also conducting over the next couple of months a consumer acceptance analysis, and later on the project we'll be bringing these two groups together with other stakeholders to identify solutions and recommendations for supporting brand owners towards market uptake of bio-based products. So I'll just turn to the survey results, and I'll come back to some of our other activities in a little while. Now, I'm not going to include all the questions because of time limitations, but just to give you an overview of the types of sectors the brand owners participating were coming from: agriculture, forestry, chemistry. We're looking at presentation, and then we had participation from biopharmaceutical, nutraceutical, construction, and energy sectors. We identified 72% of surveyed companies as already including some bio-based content within their brand owners. That's based on the CEN definition, and there would be a presumption that because there was a high representation of food companies, that's probably why that number was so high. But we asked the other companies who don't currently include bio-based materials within their branded products if they would consider to do so, and about 85% indicated that they would consider doing so in future. We turned to packaging. Currently, only 29% of the brand owners we surveyed currently utilize bio-based packaging on their branded products, but a very high number of 95% of those who don't would consider bio-based packaging in future. So this was quite a popular opportunity area that came out of our survey, was the area of bio-based packaging. And it came out again when we looked at the main categories of products that organizations would consider integrating bio-based ingredients within. Packaging at 64%, which is well ahead of everything else. And then we had smaller numbers from areas like food and flavor, fertilizer, pesticides, personal care and cosmetics, construction materials, and animal feed. One of the main focuses of our survey is to look at the barriers and risks for brand owners switching to bio-based, as well as the motivations for doing so. So some of the main barriers to bio-based product or ingredient uptake within the brand owner organizations, unsurprisingly, the expense of this was considered one of the main barriers. But we also noticed a big barrier around the functional performance and the uncertainty around this, as well as concerns about the compatibility within existing processes. So clearly brand owners are looking for something that fits quite seamlessly within their current process and their current products, and they know will reliably replace an incumbent both from a process and a product point of view. We also looked at some of the main risks associated with biobased product uptake. Again, poor functionality and incompatibility with existing processes were identified as key risks for brand owners. Lower on the list were things like uncertainty around future regulation, feedstock or ingredient supply uncertainties, as well as insufficient consumer demand. And when we looked at the specific needs of brand owners, we noticed that competitive price was very high at 83%, superior functional performance at 65%, but in some of the feedback we got through the interviews, they also indicated that the performance doesn't necessarily need to be superior. A similar functional performance to an incumbent product would be adequate for them. Compatibility with existing processes at 63% and superior environmental performance at 58%. Also, reliability of supply is seen as important, as well as clear details that they can pass on to their consumer about how the product could be managed at the end of life. And then we looked at some of the main motivations for brand owners switching to biobased. And the two standout motivations were meeting company sustainability targets, as well as meeting consumer demand, both in the 60s. We had meeting existing regulations and anticipated regulatory changes, both at 22%. Green marketing, quite high at 39%. Only 5% of brand owners cited high price or high market price for green products as being the main motivation for them switching to biobased. Other motivations included creating key selling features within their products, demonstrating that the company continues to be innovative, just beyond merely taking up a biobased product, showing that the company is really innovating, and also the possibility to deliver products that can achieve eco labels. And we asked then, in your experience, what factors are the main drivers for customer growth or growth of customer demand for biobased products? And overwhelmingly, the brand owners suggested it was around customer preference for products with environmental impact, as well as improved customer awareness about biobased products. So obviously, there needs to be some more effort to educate society about biobased products and the opportunities, whereas areas like greater availability of biobased products and more cost competitive products ranked lower. One of the things that came out of the interviews was the importance now of young people and millennials in influencing older people in households, and how that could be a game changer for uptake of biobased products among consumers. And we also asked how they foresaw their own customer demand for biobased products over the next 5 years, with 75% of brands envisaging strong or moderate growth of customer demand for biobased products over the next 5 years. That's a snapshot of the results. There's a lot more questions, but just due to time, I want to turn a little bit and talk about a couple of the activities that we're going to be undertaking within the project. But there'll be more results from this when we release our report later in the year. So using the results from our survey and using the results for our consumer acceptance analysis, we're going to be bringing together key stakeholders like customers, brand owners, policymakers, and biobased industries for regional and European level workshops to co-create solutions and co-create opportunities for better uptake of biobased products among brand owners. And that's going to happen towards the early part of next year. There's also going to be a range of knowledge transfer webinars and brokerage events that are going to be organized to try and build value chains among some of the key actors later on in the project also. And we're also going to be collecting and showcasing and promoting best practices of experience where previously brand owners have already switched to biobased. So successful case studies which could inspire new brand owners, future brand owners to make the switch and transition to biobased. And we're also going to be developing and testing tools within the BioSwitch project, validating these tools with brand owners that can support brand owners in the switch towards biobased. One of these tools is the BioSwitch self-assessment tool, which will allow brand owners to assess at which level on the switch to biobased they're currently at, and to access tailored supporting materials on the basis of the supports that they require. And another tool is the carbon handprinting tool. So this is the BioSwitch Sustainability Assessment tool, uses the carbon handprinting methodology to understand and communicate the sustainability benefits of switching to biobased. So it's based on the life cycle assessment methodology, but whereas with LCA, the focus is normally on carbon footprinting and all of some of the negative impacts of all the process, the accumulated process steps, all the negative impacts on the environment. Carbon handprinting takes a baseline system and compares it with a new product system, and it focuses on the difference between the two and looks at the positive impact that can be made by introducing the new product system to the market. And this is called the carbon handprint, and the focus is on trying to make your carbon handprint as big as possible. It's a really, from a brand owner point of view, it's a positive message to be able to explain to your customer about the benefits that the product can have, and by purchasing this product, you can actually reduce your own personal carbon footprint. So it's been tested in a number of different areas already, the carbon handprinting methodology, but specifically within BioSwitch, we're going to be validating it with flagship brand owners and testing it and seeing how impactful it is from a brand owner perspective to be able to use the carbon handprinting methodology. So that's a little bit about our project and some of our results. These are our partners, and you can find out more information at www.bioswitch.eu or contact us at info@bioswitch.eu. And these are my contact details in case anyone wants to get in touch. And over to you, Iris.
Thank you very much, James, for this really interesting study. And I have actually a lot of questions I would like to discuss with you. And I saw that you will be available for more questions also. But maybe since I don't see a question in the chat, I have one question that I find very interesting for me as somebody that works or provides bio-based resources. You talk about bio-based content, and a bit about what that is exactly. You mentioned packaging, for example, and then I saw that in the food sector, only 20% or so of the people would put more ingredients. So how do I understand that? Because I understood food is anyway bio-based, or anyway. So my question: what do you mean with bio-based content?
Yeah. So we use the CEAN definition, and basically this is a little bit of a challenge for us as well, because we noticed that from our question about how many companies already include bio-based content within some of their bio-based products, the answers were pretty high. So for us, the bio-based content was any product that contains materials coming from organic materials from the agri-food, forestry, marine sectors within their products. And then obviously food would be considered part of this. So the fact that we had quite a few food companies participating within the study would kind of skew that a little bit, where a lot of them would have viewed their food products as being products containing bio-based content. And from that point of view, that's why we think that figure is quite high when you compare it with the packaging figure at 29%, which is relatively low compared to the amount of companies who were including bio-based content within their products.
Mm-hmm.
Yeah.
And maybe 1 minute left to answer the question: when is the BioSwitch done in your view? If a company is 100% bio-based, or?
Oh, I don't think so. I mean, I think we consider a partial switch to bio-based as a switch to bio-based. So a bit like what is a bio-based product? I mean, it's typically we understand it as not necessarily having to be a 100% bio-based. We interpret it as being partially or fully bio-based. What we're hoping to do, Iris, is that if a company is already introducing partially bio-based products, if there's an opportunity to support them to increase the bio-based content of their products, that's what we'll be doing. But also there'll be a key focus on the product of the project in supporting companies who haven't actually joined the journey to transition to bio-based, and really spreading the message to those brand owners who haven't started that process about the opportunities, and showing them case studies of other companies who've already made the switch. So it's about growing in the interest among brand owners towards bio-based products as well.
That's a very important task, and I wish you good luck with that, hoping that more companies switch to bio-based.
Thank you.
Thank you very much, James, for
Thanks,
your...
Iris.
Okay. Yeah, it's also time and an opportunity to go to the next presentation. And I would like to introduce our next speaker, Wouter van Winden. Wouter holds a master degree in process technology, which he gained from Wageningen University, and a PhD in the field of metabolic engineering of microorganisms, which he obtained at Delft University, both in the Netherlands. And since 2007, Wouter has been working at the DSM Biotechnology Center in Delft as fermentation scientist. He's also involved in development and large-scale implementation of new fermentation processes, as well as in the optimization of existing ones. And he currently holds the position here of the principal scientist fermentation. And Wouter, we are very curious to hear about your 0 concepts in bioprocessing. And now the floor is yours.
Yeah, thank you, Iris, for the kind introduction. This morning I'd like to introduce to you the so-called 0 concepts in bioprocessing, which is really pushing for the limits of bioprocessing. And I'll be having this presentation on behalf of DSM, which is a Dutch-based multinational company that has by now over 150 years of experience in modern biotechnology, and in which company we are still looking at the future challenges for bioengineering, because we've been selling biotechnological products for 1.5 centuries now, but we still see, of course, a huge range of opportunities for future bulk chemicals to be produced with biotechnology as well, replacing the fossil-based chemicals that are still around today. So this presentation is inspired by two papers in literature, in which we introduced conceptual design as an important tool to come to the future bioprocesses, because we still see that after all this time of biochemical engineering, so the modern biochemical engineering, I would say, has been around for the last 50 years or so, but there's still major hurdles to be overcome to become really sustainable and really competitive with the fossil-based products. And in these papers, we've been introducing a couple of conceptual ideas where we think we need to make really radical steps still, rather than the incremental improvements to come to a truly competitive bioprocesses. So to make tomorrow's bioproducts needed by mankind, we think that we need to become more sustainable in terms of becoming cheaper. So typical development projects need to become 10 times cheaper to execute. We need to deliver strains and processes that are closer to the maximum possible. We just list here, based on literature, a couple of champions in terms of titer, in terms of productivity, and in terms of yield, which are the 3 main KPIs for typical bioprocesses. We believe that we really need to push for those limits to get competitive bioproducts on the market. Well, these products have to be, and the processes have to be developed faster than they are nowadays, and they have to be introduced in the factories on full scale with less problems than nowadays. Because on paper it's always easy to do, but having experience in implementing and introducing and starting up real plants with these bioproducts, you know that there's always a lot of surprises coming up when you start doing it on the full scale. So as said, we have introduced in those papers I mentioned in the beginning, we have mentioned the so-called 0 concepts. So 0 concepts is really aiming for the ultimate performance in a number of respects, and we've coined 8 of them. So we think that we need to aim for the ultimate performance in terms of 0 carbon spills, 0 energy spills, 0 water spills, 0 pH swings, 0 oxygen use in fermentation, 0 nitrogen use in fermentation, 0 process variability, and 0 development time. Because of time limitations today, I can only shortly touch upon a couple of them as illustrations of these concepts, but I cited some literature on the slides so people can look for more details if interested. So just as an example of 0 carbon spills, so this picture shows going from the bottom right via the whole value chain to the top left, where we have the product. The value chain going from sunlight via crops through, in this case, hydrolysates, sugars, fermentation, and downstream processing to your final product. And all along this value chain, there's carbon spills in every single step, which cost sustainability. They create more ecological impact of the product, as well as introduce costs. I just mentioned on this slide a number of ideas all along the value chain where you can reduce the carbon losses. And just to highlight one of them is an example of the fermentation where I'm active myself. So one of the major carbon losses actually in fermentation is that you waste a lot of carbon on forming the biocatalyst itself, the cells, which you have in single use and then discard again. So just as an example, this comes out of mammalian cell culture. We've been developing a process in the past with cell retention cultures, where the cells are not just washed out in these continuous perfused cultures, but where actually with a filter device, the cells are kept in the culture and kept productive for a longer while, adding to the productivity of these fed batch cultures of monoclonal antibodies, tremendously boosting the output of these processes, really making this step change rather than an incremental change to the output of such cell cultures. Yeah, then zero energy spills. Well, just one example out of industrial biotechnology. One of the energy wastes in biotechnology is energy needed to keep your processes monoseptic. You want to keep only your production organism in your vessel. And on large scale, this is really a major challenge to do this. And of course, we can spend a lot of energy to keep everything heated, to fully sterilize, heat sterilize the media and all the feeds, wasting energy to the environment. So there's alternative conceptual ideas for that, which could be the use of extremophiles. So you go to fermentation conditions where competing organisms simply don't make a chance to overgrow. An alternative is to use even xenobiotic nutrients. So you engineer your organisms so they can use one of the nutrients that are not commonly used by the natural competitors, saving this energy for sterilization. Now, of course, we're looking at the full value chain, so not just focusing on fermentation. So you have to look at the downstream processing as well. A lot of energy can be saved by looking at the production of water-insoluble products, because separating water-soluble products from the fermentation broth often comes with a huge energy cost. So if you can make gaseous products, like here in the biogas example, if you can make apolar, so lipid products, or extracted in the lipid phase, like we do here in our own site in Brotas in Brazil, producing farnesene as a product, or if you produce products that are in the solid form or in crystal form in situ, like we do with our vitamin B2 production in one of our plants, that is a tremendous energy saver downstream. So you really have to look at this integral bioprocess design for that. pH swings, just an example out of our own developments, succinic acid, where if you look at literature, a number of companies have been developing bacterial processes where you need to ferment at neutral pH. So that means you have to titrate a lot of salt to keep your fermentation at neutral pH, and then in downstream processing, you need to go to low pH to recover the actual acid. We developed an yeast-based product process where we went to a pH below the pKa of succinic acid. So no titration was needed. The fermentation was done at low pH, and the separation as well. So you don't produce all those salts, and you don't need the complex downstream processing to remove all that salt again, or to even recycle them. You can look at zero O2 processes, anaerobic. That's the ultimate yield often for bioprocesses. And then you have to look at literature to see, you have to track actually the Gibbs-free energy that is present in the electrons in your substrates and the Gibbs-free energy of your products. And you have you can look then for combinations of substrates and products which allow you to produce your products anaerobically and thereby normally with a much higher yield. Zero water spills. All the water you introduce in your process has to be removed at some point. So looking at the feedstocks of your fermentation processes, you have to try to introduce as little water as possible. Well, the typical glucose syrups, so as we call them first generation carbon streams, they contain about 40% of water. The second generation, so the lignocellulose-based hydrolysates, often even more water. So we're looking at whether you could use gaseous substrates, and in the end, maybe just CO2 and renewable electricity as substrates, where you really don't introduce any water in your process, so you also don't have to remove it anymore. And an example of this, and then I come back to this the Gibbs-free energy. You can see in this diagram that, for example, carbon monoxide and hydrogen are excellent anaerobic substrates, and they can produce ethanol, which is much lower on this diagram. An example of that is the LanzaTech technology, where gas substrates without introducing any water can be used to produce ethanol from flue gas of steel industries. Yeah, and the final example of a zero concept is zero development time. So what we're doing, and this is the point of not implementing your process on large scale and then having all the scale up problems, that the performance on a large scale turns out worse than on a small scale. What we try to do in our development is really model the large scale fermenters at the earliest phase of the R&D projects. You need combined CFD and metabolically structured models for that, with which you can simulate the lifelines of individual microbes, and you can see how microbes experience life in a full-scale fermenter at, say, 100, 200 cubic meters. Those conditions we try to downscale as good as possible to lab scale or even micro-scale reactors to really mimic those large-scale conditions, and we develop our processes and we select our strains under those conditions, so that the conditions and the organisms that we then develop on lab scale, we know beforehand that they should perform well on full-scale conditions. So that ends a very short overview of these zero concepts, where we, as said in the beginning, we think we really need those radical changes, going for those very ambitious targets to come to the truly sustainable and low-cost bioprocesses of the future. Conceptual design in that is really an essential stage for success. So really starting with a good design over the whole value chain. So we see grand challenges on the way forward for the bioprocess future, but we truly believe that that once we can reach those goals, then fossil-based production will be something of the past. So with that, I would just like to acknowledge some inspirations of this talk, and I'll be happy to take any questions there are. Thank you.
Thank you very much, Wouter. This is what I learned is very important in making bioeconomy happen, is that you also have the technologies to improve efficiency and to reduce greenhouse gases. And I think you showed a broad range of technologies and approaches here. Thank you very much for that. I'm afraid that going into different technologies, we don't have the time by now. So and as I don't see an immediate question in the chat, I would think maybe there are some people that have questions on the technologies, but you offered kindly that you would be available after this session for some discussion and for some questions. And
yeah, sure.
yeah, my suggestion would be to move the discussion to there, because I don't think we have enough time to go into technologies now. So thank you very much, Wouter, for the presentation, and hope for a good discussion later.
Okay, thank you.
Then I think we will come to the last of our 4 presentations. And the final speaker in this session will be Jennewein. He studied chemistry, pharmacology, chemistry at universities in UK, Switzerland, Germany, and also in Germany. And after working at Washington State University,
Hello? Hello?
so I'm back again as moderator, but I think it's a turn on Stefan's presentation now. Please, can the organizers make sure that he can present now? Okay.
Yeah, thank you very much. Hope you can hear me. Somehow the line broke, but hello, good morning. My name is Stefan Jennewein. I'm founder and currently CEO of Jennewein Biotech, a company which we actually are currently selling to Christian Hansen after more or less pursuing the field here of human milk oligosaccharides the last 15 years. I don't know whether you came across about us already in the past. Jennewein Biotech is a biotech company which we founded back in 2005, focusing on the manufacturing of complex oligosaccharides and monosaccharides. What we do is simply like we combine innovative biotechnology with chemical engineering and mainly also like metabolic engineering to construct fermentation strain for our sugars, which we then ferment in large scale. So Jennewein is running like fermentation on scales of 100 to 200 cubic meters, like probably several of them every week. And we are producing like these mono oligosaccharides in hundreds of kilograms to multi-ton scale as spray dried and also like a crystalline product. Early on, I also had to find out that that does not simply is sufficient for run a biotech company. So we actually also had to look in worldwide registration. So we were the first here in Europe filing on a novel food application on a product which came out of a recombinant bacterial process. We were also one of the first in the US filing registration of like fermented products for infant food. We do worldwide marketing. We also run our own tox studies and also clinical trials. Recently, we just completed a large multi-center clinical trial in Spain, Italy, and Germany for a new product. We also market our product, and I think Jennewein is a company having more or less everything what it takes to get a product from an idea to a marketable product on the shelf of a supermarket. So we are headquartered in Rhein-Breitbach, that's close to Bonn, where we since 2007. And since about 3 years, we also have an R&D center, which also houses our IP department in Bonn. And more than 1.5 years ago, we also bought the Artus Mineral Spring in Bad Hönning, which is close by, which will be now transferred into a further production plant for our human milk oligosaccharide range. So why did we get so excited about human milk oligosaccharides, even that oligosaccharides are not like a very popular class of natural products? It was simply the fact that we found out that like more than 100 years ago, somebody called Theodor Escherich was getting excited about a quite surprising discovery that infants which were breastfed at that time had about a 7 times better chance of survival than bottle-fed infants. And he was curious what might be the basis of this difference, or significant difference. And he found that there's a difference and made this difference, and then simply found that in human milk you have complex oligosaccharides which don't really nourish the infant, but have multiple physiological effects beyond providing nutrition. And these are simply missing in other milks of other mammals, and that led simply then to the discovery of several HMOs over the next 100 years. And just let's have a look on how human milk is composed. First of all, human milk is a product which is around for several million of years since the beginning of human mankind. And human milk oligosaccharide constitute the largest, or the third largest fraction of human milk besides lactose, lipids, and then we have the human milk oligosaccharides. This human milk oligosaccharides is not a single compound. It's an array of at least like 15, 20 major compounds, which you can actually identify easily by HPLC. And the major, in 80%, the most dominant HMO is 2-fucosyllactose, and that was also the first product Jennewein Biotech introduced to the market in 2015. As said, like human milk oligosaccharides is not something which provides energy to the infant, but has physiological effects. The most intriguing effect, and that was also why we got so interested in human milk oligosaccharides, is simply like they act as decoys and can significantly reduce infectious diseases. That's not only for bacteria, but also for viral. It also binds toxin. So about 80% of all human pathogens you can actually address with human milk oligosaccharides. They are also more or less beneficial for reducing inflammation. It's something like which I don't really want to go into in too much, but some further effects are like our reduction of allergy risk, and that's simply coming in also with the prebiotic and microbiome effect. And another very beneficial thing which they bring on the table is like brain development, and that mainly comes to the scarce monosaccharide and fructose, we think, and that's also some products which we manufacture besides the human milk oligosaccharides. Just elaborating a little bit about why we actually pursued human milk oligosaccharides or complex oligosaccharides in general. It's simply like, as you might know, all cells are glycosylated. These structures serve as receptor or co-receptor for nearly 80% of all human pathogens. And simply by providing soluble oligosaccharides, you have decoys, so pathogens bind preferentially to these decoys, which are also there can be there in higher concentration, and simply like you interfere with the more or less most fundamental process of infection is simply like the binding of the pathogens to the human cell, and by this you simply confer a protective effect. That's something like which is widely shown or over the last decade on like bacterial infections, like also some of our own work addressing things like Campylobacter, Pseudomonas, and or EHEC, like pathogenic E. coli. You can simply inhibit with these oligosaccharides. And about 5 years ago, we even engaged into viral diseases. One thing which we think is one super beneficial claim, especially for infant food products, is like inhibition of norovirus. There we started a larger programs also involving academic collaboration. And here we could already build on that. It was known that like breastfed infants have lower incidences of norovirus infection. And we could then, in collaboration together with Grant Huntsman from the DKFZ and Professor Schroten and his team from the Kinderklinik in Mannheim, simply show how this inhibition function works. Is simply like that 2-fucosyllactose, but also 3-fucosyllactose can bind to the P protein of norovirus and simply interfere with the infectious cycle. So, as said, like Theodor Escherich established by his work, the foundation of what is today known as pre and probiotic, and HMOs simply have 1 million years of co-evolution with beneficial bacteria. So it was simply shown that infants' microbiome simply contains like bifidobacteria, which can chew on the HMOs. And so HMOs are quite essential of building what we say a healthy microbiome in the first, like 1,000 days of life, or as long as kids are actually breastfed or consuming infant food products. And still today, formulas not containing HMO differ quite significantly from breastfeeding. So I don't want to go much into details on the right side of the slide, but you don't have to be a microbiologist that just by eyeballing here the circles, you definitely see differences in the distribution and also species which are involved in establishing the microbiome. And there's huge differences still today existing between like bottle-fed and breastfed infants. The microbiome became over the last 5 to 7 years a major area of interest, because microbiome is today really related to a lot of very complex diseases. Like probably most obvious is like obesity. Obesity is already a huge problem even for small children, like in societies like in the US, but also China. About 30% of small children are already obese, and that's definitely not something which is a genetic trait, but something which is definitely based on the microbiome and the nutrition. So this is an area of high interest here also for us at Jennewein, and we currently also are just completing a big clinical study, multi-centered here in Europe, which we ran with more than 300 infants in Spain, Italy, and Germany, where we also looked into like the microbiome of this first 4 months of life, and how differences in the microbiome actually originate out of like breastfeeding. It's also not all moms contain all HMOs; they are quite individual. And on a new product which we developed, which is called HMO Mix of 5, and simply standard infant food formula. More or less coming to the nuts and bolts, what Jennewein is actually doing is like Jennewein is first of all a manufacturer of HMOs. So we were the first who actually could manufacture HMO on large scale. We managed that through simply biofermentation and smart technology, and how we could simply cost effectively purify these oligosaccharides out of fermentation, and we were able to launch that initially back in 2015 in the US, and then later here in Europe, and I will later elaborate a little bit on that. Our HMOs are more or less sold today worldwide. The first product is 2-fucosyl-lactose, which we launched in 2015, which was also approved in most countries at concentration of 1 to 2 grams a liter, and was initially used at a much lower rate of about 0.2 grams a liter initially, because people thought that we were not able to scale that into volumes that could be simply provides enough supply for actually natural concentration use, but that actually proved wrong, and today we are going to natural concentration with this HMOs in infant food products. A second product which was then introduced in the market last year is the combination of 2FL and neo-LNT. Especially I would like to return to US in 2016 with the first product, and then became a huge success worldwide. And I think about 90% of all infant food products are today sold in the US contain 2FL. It's also then became a huge success. We were then able to introduce that in the European market, where we still are less infant food formula, just catching up with HMOs. Became a huge success also in Southeast Asia, also in sales into China via Hong Kong. It's I have to tell you that probably 60 to 70% of infant food formula in China are sold via the internet. So we are still working on the approval of 2-fucosyl-lactose and also other HMOs in China, but through to this cross-border sales via Hong Kong. And so it's these infant food formulas are already on the market. I think today we sold, or with our partners or customers, we sold probably already 500 million of packages of infant food formula containing our 2FL. Jennewein also saw very early on the need to establish new manufacturing technology, especially for oligosaccharides. There are not many biotech products which are falling into the class of mono and oligosaccharides. That was simply an area overlooked, and we started already back in 2013 investing in manufacturing or production capacity for oligosaccharides, which is a little bit a niche technology using like membrane processes, electrodialysis, and so on, and also in large-scale fermentation. So we have like fermentation capacity in Breitbach, but also because of market demands, we also had to outsource quite significantly fermentation capacity here throughout Europe. Currently, we are introducing two new products. One is a product which is on customs demand, and the second product is the HMO mixer 5, which contains the most abundant neutral HMOs like 2FL, 3FL, neo-LNT, 6SL, and 3SL, which are silated HMOs, and we also managed to introduce that into the market at a natural concentration. This leads to simply like at the moment a massive expansion of our production capabilities, because we are simply like at the moment bringing up volumes about 30-fold by switching from 2FL to this 5-HMO mix. It's like, I would say HMOs, and in particular 2FL, was a really success story, and also a success story for Jennewein, because we started it out back in 2005 with a simple idea that we want to, with biotechnology and metabolic engineering, come up with a significant large bioproduct, which you can also sell in the range of hundreds to thousands of tons. And at the very end of the day, I think we are one of the very few biotech companies which actually could put their idea and their concept into reality. So in 2016, we initially launched with Similac, which was the #1 and still the #1 brand in the US with 2FL. As that is a concentration of 1.5 grams a kilogram. And currently we're introducing, or this will go on market early next year in at least 2 world regions, a product which is called 5-HMO mix, where we actually brought up the concentration about 30-fold in the product, going from 1.5 grams to more than 40 grams a kilogram final infant food formula. So this is becoming now a reality early next year, and currently the company is already looking into the next step of innovation, which is more or less the replacement of GOS and FOS. That means that we are ramping up concentration even further, going to like at least 60 grams a kilogram. And as you see, I put at the far right the natural concentration, which is about 15 grams. This also varies during the course of lactation. We can start up with concentration as high as 25 grams a liter, and going then to lower concentration as long as more or less lactation is practiced. So there we have it still in a natural concentration of about 100, more than 100 grams a kilogram. So it's like the next innovation Jennewein is working on is more or less the replacement of GOS and FOS. That also means getting from like volumes really ramped up even further than we are doing today, but also getting production costs into like price tags, so that we can also compete with GOS and FOS on a market price. So I more or less at the very end of my presentation, and I would like to hand over back to Iris. Yeah, thanks a lot.